Good morning. We're going to get started with our next session, Environmental Toxins and Cancer. My name is Farah Khan. I'm a medical oncologist here at the University of Vermont Cancer Center, and I'm happy to introduce to everyone Dr. Larry Engel, who will present this morning's session. Dr. Engel's research focuses on the impact of disasters, including oil spills and hurricanes, on a range of health outcomes, pesticides, persistent organic pollutants, and hazardous air pollutants in relation to cancer and neurological deficits, and occupation of environmental risk factors for chronic kidney disease. He leads the program in environmental and occupational epidemiology in the University of North Carolina Department of Epidemiology. He teaches a graduate course and lectures on environments, occupation, and disaster epidemiology. This is a very interesting topic, and I anticipate a great deal of questions after Dr. Engel's presentation. For the Q&A at the end of the session, for Zoom attendees, please chat in your questions, and a volunteer will read them aloud. For in-person attendees, if you have a question, please wait to receive the microphone to ask your question so that our colleagues on Zoom can also hear your questions. And now I'll turn this over to Dr. Engel. Thank you.
All right, thank you all for coming today. This is a topic that's kind of near and dear to my heart, so I will try to stay on time. It can be challenging, but I have a lot of material I want to talk with you about today, and I hope that you will find it informative, and I welcome your questions afterwards. So the main topic today is environmental toxins and cancer, as the title indicates. But what I wanted to do was take not just tell you what I think are environmental toxins and cancer, but to talk to you also, to talk some about that, but also talk about how we figure this out, how we make those determinations, and some of the challenges that are inherent in that, and why it may feel like progress is really slow and that there's so much more to be done. So hopefully, at the end of the talk today, you'll have a better sense not only of environmental carcinogens, but also of the progress that we're making and how we're doing that, and some of the obstacles that we face. So this is the general structure of what I'll be presenting today, just first starting with just some trends in cancer rates, both incidence and mortality, talking about some known or established carcinogens, some of the challenges in studying environmental carcinogens in particular, and then I'll give you a couple of examples from my research that will illustrate some of the points that I hope to convey today, and then we'll wrap up. Can everybody hear me okay? Good, okay. So first, just some background. Many of you may have seen these numbers already, but the first point I want to make is that, as we are well aware, that as this table shows, on the top, cancer incidence, on the bottom, cancer mortality, that the leading cancers today in the United States are prostate cancer among men and breast cancer among women, and they by far lead the other cancers, the next most common being lung cancer in both men and women, and colon cancer. And the rates the mortality rates differ in that lung cancer dominates for a number of reasons, because of detection, treatment, and other factors. But prostate and breast also rates remain quite high for across the country for these cancers. Make sure I don't forget anything. So let's take a step back and look also at cancer trends over time, since that's certainly very relevant for an environmental focus. So what I want you to note here, a couple of points. One is that we'll start with the cancer incidence in women, since it's a little bit simpler of a story. The cancer rates have been going up in women over time since these data began in 1975, and they've steadily increased over the last 45, 50 years. The story is a little bit more complicated in men. You see a big surge here in the early 90s, and then a quick drop, and then a continuing gradual drop. Since then, although it's stabilized, and it's only slightly increased in men from what it was 45, 50 years ago. The reason for this surge here in men is largely because of the rapid uptake and widespread uptake of PSA testing in the early 90s, which led to a big surge in the detection of prostate cancer. That has largely subsided over time, but the rates are still a little bit higher today than they were in the 1970s. But this only tells you part of the story, because this is all cancer, and it masks a lot of important detail that is important to recognize. So let's look at individual cancer types, which I think is illuminating. The first thing you notice is that, we'll start with men since they're on the left. Again, as I noted, the surge in prostate, the big increase in overall cancer in men, it was driven largely by the surge in prostate cancers starting in the early 90s. But what's worth noting here is that, and this is incidence, by the way, cancer incidence. You'll notice that a number of cancers actually have gone down in rates over time. So lung cancer has steadily decreased since the 1990s in men. Colorectal cancer has also decreased over time. Certain cancers, on the other hand, are increasing. So melanoma rates have gone up over time. I know that's a particular concern here in Vermont, but it's happening elsewhere in the country as well. Liver cancer also has slowly increased over this time period. Now, in women, what you'll notice is that the increase in cancer that you saw in the previous slide in overall cancer is really being driven largely by breast cancer, which I think is something that we've recognized for a while. But again, there are some bright spots in this picture that lung cancer also has been decreasing in women, not for as long as it has been for men. Women took up smoking later and they stopped smoking later. But cancer rates have been decreasing in women for the last 20 or so years as well. Colorectal cancer rates are also down in women. But again, there are certain cancers that are increasing in addition to breast cancer. You see an increase in thyroid cancer, you see an increase in liver cancer. So the picture is really mixed depending on which cancers we're talking about. But there's still clearly, there's a lot of progress that we've made, but there's obviously a lot of work left to be done. But the last thing I want to make on this topic is also that cancer incidence is one important measure of the cancer burden in the population, but the mortality is also critical to look at. And on this note, the picture looks encouraging. It's not where we want it to be, certainly, but it's certainly improving that for both men and women, cancer rates have been going down for the last 20 or so years. So cancer mortality rates. So we're making progress there, but clearly we need to reduce incidence of cancer. We need to improve treatment, detection and treatment of cancer. But certainly there has been a lot of progress made on this front. Now, the one other thing I will note here is since I'm in Vermont, you're mostly in Vermont already, is what the cancer picture looks like in Vermont. And here, I just want to note, this is cancer rates. On the left, you see the cancer incidence across the entire United States by the darker blue, the state, the higher the incidence of cancer, overall cancer in that state. On the right, what you see is the rate of change of cancer in that state. And what you see for Vermont is that while you're not as low as we would hope, you're in the bottom half of cancer incidence for the country, and interestingly, you're much lower than most of the surrounding states, the rest of this region. And encouragingly, your cancer rates are going down faster than many other places. So there's still work to be done, but things are moving in a positive direction for at least some cancers here in Vermont as well. So let's change to the focus of today's, primary focus of today's talk, which is environmental carcinogens. And first I wanted to start with a broad overview of cancer risk factors in general, because it's necessary to start there before we start talking about the environmental component of that. And apart from genetic factors, which we won't talk about today, it's important to recognize some of the behavioral or lifestyle factors that are contributing to the cancer burden that we see in the U.S. and elsewhere today. And I've listed a few of these here. It's important to note that some of these are behavioral, although behavior is a result not only from choices that we make, but from the opportunities that were presented as well. So some of these are not complex, are due to complex social dynamics. And one of these in particular, obesity, is one that I think we've gained an increasing understanding of over time of the underlying drivers of the obesity epidemic we have in this country. And there's a lot of interest and a lot of research being done on environmental carcinogens, obesogens, looking specifically for factors that might be in the food we eat or the water that we drink that may be exacerbating the obesity problem that we have in the country, that may be contributing to obesity. The last point I want to make here is that I'm not going to leave you today with a percentage or a number of cancers that are caused by the environment, because no one can agree on what that number is. And it's not just industry versus scientists. It's actually scientists versus scientists. It's a really difficult thing to quantify, because it's difficult to quantify the environment. It's difficult to quantify what the risk is, and it makes it very difficult to put a number on it. And so, for perspective, the numbers are anywhere from about 2% to about 90-something% that people have estimated. So essentially, we can't give you a number. But suffice it to say that we know that environment is an important contributor to cancer, and it's important that these are modifiable factors that we can address and that we can intervene on in order to reduce the cancer burden. So let's talk briefly about some established carcinogens. These are carcinogens that various national or international organizations have identified based on the weight of evidence as carcinogens. And there are two different formulations or classifications that I will present today. One is from the National Toxicology Program, which is a component of the National Institutes of Health. They have identified 63 carcinogens known to be, agents known to be carcinogenic in humans, and 193 that are reasonably anticipated to be human carcinogens. The distinction there being that the weight of evidence is not sufficiently strong, according to their classification, to categorize them as known carcinogens, but the evidence is strong enough to deem them as of concern. And the other source is the International Agency for Research on Cancer, also known as IARC, which is an agency within the World Health Organization, which has identified 129 Group 1 carcinogens. They use a different nomenclature, but the idea is similar. Group 1 carcinogens are known carcinogenic to human. They've identified 96 Group 2A, which are probable human carcinogens, and Group 2B, and 321 Group 2B carcinogens, which are possible human carcinogens. And again, this comes down to the weight of evidence. And these can change over time. They reevaluate periodically these agents, and sometimes agents can move up or move down in their classification based on additional evidence that is accrued over time. The one last thing I'll note on this slide is that these are not all environmental carcinogens. These include a range of agents. It could be microorganisms, it can be behavioral factors like shift work, as well as drugs and other agents. But the topic here today is environmental carcinogens. So let's talk about that. In the U.S., over 86,000 chemicals are registered under the U.S. Toxic Substances Control Act, which was first passed in 1976 and was updated in 2016. And this, under this act, about 42,000 chemicals that are registered are currently in active use. So the reason I'm giving you these numbers is to illustrate this is a lot of chemicals that we're being exposed to. We're not being exposed to every one of these, but across the country we're being exposed to many of these, different people to different mixtures of these. But nonetheless, this is a lot of chemicals that are in our environment. And importantly, hundreds of new chemicals are being introduced every year. So the number only grows of chemicals that we're potentially being exposed to. And for perspective, less than 2% of these have been what we deem adequately studied for cancer. And part of that is just because this is an enormous number. 86,000 chemicals is a lot of chemicals to study. And until the last few years, these agents were studied one by one because that was the only way that we could do it. We're now developing technologies and methods that allow us to study larger numbers of chemicals, but this is a recent development, and we'll talk more about that later, but the project has been enormous, the magnitude of the need. I guess the last thing I should mention here is I'm not suggesting that all of these chemicals are carcinogens. Many of them are probably very unlikely to be carcinogens, but we don't know because the number is large and a lot of them have not been studied. There's a lot there, I think, to uncover. And then another statistic that I will give you is on the amount of hazardous substances that are released into the environment in the U.S. every year. So there is a registry known as the EPA Toxic Release Inventory, which requires industrial producers of chemicals deemed to be toxic to report the amount that they release into the environment every year. And in 2022, almost 22,000 facilities were reporting to this toxic release inventory and reporting 3.3 billion pounds of chemicals that were being either released or disposed of over a single year. And this number, for perspective, this number has gone down since these data started being collected in the 80s. So it's, again, the amount that we're being exposed to of these chemicals, many of which really haven't been well studied, is quite large. So I've presented here a list of environmental agents that we know are carcinogens. These are largely established carcinogens. And it's a range. They're not organized by anything other than alphabetical. And I want to make a couple of points here. One is that these represent a combination of natural and man-made agents. Aflatoxins, for example, are a toxin that's produced by mold and that occurs in a number of different foods. These are naturally occurring, although the way that we grow and store food affects the levels that we're exposed to. But a number of these are also naturally occurring. So, for example, UV radiation, arsenic, which is found in geologic formations in groundwater throughout the world, but is more concentrated in some places than others. And then a number of these are man-made. So, for example, or largely man-made, such as air pollution, and chemicals like benzene, which are primarily anthropogenic. And another point to note here is that the routes of exposure to these vary widely as well. That some of them we get through food, some primarily through water, some through breathing. Some of them are ubiquitous, such as air pollution. Some of them are structural, primarily such as asbestos, which will be found in our built environment. And then the last point here is also that we've identified links between environmental agents and a wide range of cancers. So I won't go through all these, but suffice it to say that most cancers have been linked to at least one environmental agent. But an important note here is that many of these exposures we are unaware of. So most of these, at the levels that we are exposed to them, we can't taste, we can't see. So it's an invisible exposure that goes on all the time. But I want to shift focus now to talk a little bit about the methodologic challenges that we have in studying these types of relationships. And because I think that, sometimes we look back and we think the war of cancer has been going on for over 50 years now. Why haven't we solved this? Why haven't we identified all of the environmental carcinogens? And part of it is just the sheer magnitude of the question that, again, with 86,000 chemicals that need to be studied, that's a formidable task. And so there are challenges just in terms of the sheer scale of the question, but there's also some particular challenges of studying the environment in relation to cancer. And some of those issues are that cancer has a long disease latency, that for most cancers, the time between exposure and identifiable disease is generally 15 to 30 years. So there's a long time that we need to be able to track someone's exposure in relation to their disease. In addition, and related to the previous point, is the lack of historical data. So when we're studying a disease that might take 15, 20, 30 years, we need to know about the exposures that occurred in the past. And oftentimes those data are not available. There are a lot of chemicals that we're studying today, for example, perfluorinated chemicals that probably you're all familiar with, for which there are no data going back more than 20 or so years. Those data weren't collected, and so it's impossible to go back and retrospectively figure out what people were exposed to, because there simply are no data to do that. And the alternative is to collect specimens and samples today and follow people for the next 30 years, but it's a long time to wait for an answer to a question that we need answered sooner. Some of these exposures many of these exposures are difficult to measure and or maybe changing over time. So perfluorinated chemicals are a good example, where for many years we were primarily exposed to two main ones called PFOS and PFOA. But since those chemicals were voluntarily withdrawn from the market in the US, they were replaced with a family of thousands of new perfluorinated compounds. So we now know how to study PFOS and PFOA because we've been doing it for years, but we're still trying to figure out how to actually even measure this wide range of thousands of other perfluorinated chemicals, and there's no historical data on those. So this is a problem that we need to solve and that we will continue to do, but presents some big challenges. There's also a lack of suitable specimens. There are quite a few prospective studies in this country now where participants were enrolled, usually healthy, and we interviewed them, we collect blood, we collect urine, we may collect hair or other specimens, and we store it, and we study these people over time, and we go back later and we measure different chemicals in these specimens that we've stored. And that works great for many questions, but one of the hot topics today is microplastics and its relationship to cancer and to other diseases. And one of the challenges that we face is that a lot of those specimens that were collected, blood specimens or plastic or urine specimens that were collected in the past, are stored in plastic tubes. So now, you measure microplastics in those specimens. You don't know whether you're measuring what was in the tube or what was in the person's body. So some of these things are problems that we didn't anticipate, and so weren't prepared to address. And so it presents, again, additional challenges that we need to figure out. We will. I'm not trying to be pessimistic here or to leave you despairing, but these are real challenges that we face. Other issues are disease heterogeneity, which I think many of you are also familiar with, that over time we have increasingly come to understand the complexity of diseases like cancer, and what formerly might have just been called breast cancer, we now recognize as multiple different diseases, each of which is likely to have a different set of risk factors. And so over time, we're better able to study those diseases, but it presents challenges in terms of retrospective analysis, looking back over time, because the data often aren't there to allow us to adequately do that. And then the last point I'll make on this slide is the issue of mobile populations. And this is one that is particularly problematic for cancer, again, because of the long latency, that we need to be able to oftentimes be able to track people over time or know where they lived over time. And the more mobile people are, and people in the United States tend to be pretty mobile, the more difficult it is to study these relationships. So now I'm going to talk about pesticides and cancer. So one of the topics that I've been particularly interested in my whole career. And I'm going to speed up because I'm taking a long time here. But what I want you to notice on this slide is that since 1990, the amount of pesticides used worldwide has almost doubled. And what's of note here, this slide shows you the use of these chemicals by the different chemical classes, different types of pesticides, insecticides, herbicides, fungicides. Well, you'll notice that most of these haven't changed that much. The increase is really being driven by herbicides. And the reason for this is that in the mid to late 1990s, genetically modified crops were introduced into the marketplace that were resistant to herbicides. The first ones being these what were called Roundup-ready crops that, whereas before these, a farmer had to apply his herbicides well in advance of planting his crops or else he'd kill his crop. Now you can spray herbicides on your crop throughout the entire growing season because those plants have been modified to be resistant to the herbicide. So as a result of this, the amount of herbicide use has gone way up, and I'll show you another slide in a moment to illustrate that point. In the U.S. in 2021, over 1 billion pounds of pesticides were applied. It represents 1,400 different chemicals that are different pesticides that are registered in this country, and I'm going to shift now to talk about to go back to the Roundup that I mentioned a moment ago. Its chemical name or generic name is glyphosate, and what's noteworthy here is that this is the first herbicide for which resistant crops were introduced. And the point I want to note here is that the use of this chemical, primarily for corn and soybean, has gone up over 20-fold since those crops were introduced. So again, the exposures have gone way up, and of course, glyphosate has been in the news quite a bit in recent years because of concerns about its association with non-Hodgkin's lymphoma, and this adds fuel to the concern about what those risks might be. So, focusing on more broadly on pesticides, there are about 40 active ingredients in pesticides are classified by IARC as probable, possible, or human carcinogens. Many of these are still in the market in the U.S. And it's important to note that this isn't just 40 different products that you can buy in the store. This actually represents hundreds, because these are the active ingredients that can be used in multiple different products, sometimes by different manufacturers. So it's actually a large number of products that actually can contain these chemicals. And one other aspect of this question of pesticide cancer risk is the issue of inert ingredients, which doesn't get much attention because it's particularly hard to study. But inert ingredients are the chemicals that are included with the product that you buy. So you might be buying Roundup, but Roundup isn't just the active ingredient. It also contains other chemicals that help the chemical to stick to the leaves that you're applying it to, or that increase its solubility in water so that you can dilute it and apply it. These chemicals are not regulated. They're not listed as ingredients on the pesticide products. So it's virtually impossible to study these in any rigorous way because we often have no idea what those inert ingredients are. And by inert, by the way, I put inert in quotes because they're not necessarily biologically inert. They're inert in the sense that they're not the active ingredient that's killing the pest that you're trying to kill, whether it's an insect or a weed or something else. So a number of different mechanisms have been identified, primarily in vitro, for a potential carcinogenicity of pesticides. And I won't go through all these now, but suffice it to say that pesticides are a complex group, and I meant to mention this in the previous slide, and I forgot to, but we talk about pesticides as a thing, but in fact, pesticides are a broad family of chemicals that are designed to kill various things. Sometimes it's insects, it may be plants, it may be fungi. But because their targets are very different, their structures and their biological activity are very different as well. And so we might expect that their unintended activities in our bodies might be similarly diverse as a result of the variation in those chemicals. So we see a wide range of potential health effects of these chemicals in humans. including a range of potential carcinogenic mechanisms. Now, most of the research that has been done of pesticides in cancer has been done in farmers. And the reason for this is that farmers offer a number of advantages over the general population for studying these things, even though there are far more people in the general population than there are farmers. But farmers have the advantage that they have, in terms of research purposes, that they have higher exposures to these chemicals than most of us do. There are still large numbers of farmers operating in this country and globally. Farmers are very stable. Many farmers spend their entire lives in the same place that they're born, so they're easy to study over long periods of time, unlike the general population. And perhaps one of the most important aspects is that farmers have a very detailed knowledge of the chemicals that they're using and what they're being exposed to. Oftentimes, many of us will just, you may get a can of Raid or Black Flag and you'll spray it for ants or for termites or spray it on or apply something to your yard, but we often don't have the same level of knowledge of what we're using that the farmers do. So that makes a big difference when we're trying to figure out which specific chemicals are a particular concern. Now, for the public, most of our use is on lawns and gardens and around our homes, and to some degree in our diet. For people who live in agricultural communities, there are the agricultural exposures, of course, that come from the aerial application of pesticides that overshoot their target, or the contamination of drinking water and other sources. But for most of the general population, we have no idea what chemicals we're being exposed to. It's, again, either we don't know what's in the products that we use, or it's in sources that we have no information about. So the research on pesticides and cancer among farmers is mixed. There are a number of cancers that have been shown to be increased among farmers, and this is a list that probably results from a number of different mechanisms. But part of it is related to pesticides, but some of it also may be related to sun exposure and other factors. But nonetheless, this is a list that's been associated with agricultural work. And interestingly, farmers also have a lower rate of some cancers. This is worth noting, in particular lung, bladder, esophageal, and colon. And this is presumably because farmers tend to smoke and drink less than the general population. They also are more physically active. So it's not, even for farmers, the picture isn't entirely dark. So now I want to talk briefly about one of the studies that I have participated in, or that I have conducted, which is looking at insecticides and breast cancer risk in the agricultural health study. Insecticides have been linked to a number to mammary tumors in a number of animal models, and not just insecticides. We often think about DDT, but actually there's a number of other chemicals, a number of other insecticides that have also been associated with or have been linked to mammary tumors in rodents. And also a number of in vitro effects that we've observed for a wide range of chemicals that are probable carcinogenic mechanisms. Now, again, much of the attention over time has been focused on the organochlorines and DDT in particular. And one reason for this was that DDT shows endocrine disrupting effects. But it's worth noting that these endocrine disrupting effects are noted in a range of other pesticides as well, not just organochlorines. So they're also noted, they've also been observed in organophosphates, carbamates, and pyrethroids, all of which are insecticides, as well as triazines, which are a class of herbicide. So we need to look more broadly in terms of the endocrine disrupting potential and possible breast cancer risk associated with these chemicals. So the agricultural health study was started in the mid-90s, and it was based in Iowa and North Carolina. And the power of this study was that it was based on recruitment through mandatory training and certification in these two states. So we have a representative sample of farmers in these two states. And the study recruited about 52,000 farmers, private pesticide applicators who are primarily farmers, as well as 32,000 of their spouses. And this study has now published hundreds of papers on cancer and non-cancer outcomes among this population. So information was collected through interview and questionnaires on a number of different factors, including extensive questions on pesticide use, of course, since that was a primary hypothesis of this study, but also on use of protective equipment, so factors that might reduce your risk of exposure when you're using these chemicals, as well as other factors that are of interest, smoking and drinking and physical activity and so on, that might modify your risk of cancer and other outcomes. So for the purposes of this analysis today, we had about 15 years of follow-up of this cohort. The cohort's now been followed for closer to 30 years, but at the time that we did these analyses, it was a number of cycles back. And we looked at 3 different exposure metrics in the women. I studied specifically the breast cancer in the females, in the spouses. And we looked at every use of 50 different chemicals among the women. Is total intensity weighted days of use, and by intensity weighted days, what I mean is we look at the chemicals that the women report using, and we also take account of the protective equipment and practices that they use that modify their risk. So we estimate their exposure based on not only on what they're using, but on how they're using it, how they're handling it, which can either increase or decrease your estimated exposure. And we also looked at the total number of days that the women's husbands were using the chemicals, because the women can be getting indirect exposures through a number of means that way. And I'm going to go just briefly through some of these results here. And I want to note also that I'm presenting only insecticide results today. We've published a number of papers on this, and I would encourage you to either ask me afterwards or look at some of the papers or follow up with me by email after this meeting. And we can talk more about this, but I'm going to focus just on insecticide use today. And what I wanted to note to you was that we did identify a number of insecticides that were related to an increased risk of breast cancer in this population. And the 4 that are highlighted in red here are the ones that stood out primarily in this analysis. Heptachlor is an organochlorine insecticide. The other 3 are organophosphate insecticides. So a different class of chemical, but again, they also show endocrine disrupting attributes. And to help, for those of you who aren't involved in research, what these numbers represent here is the relative risk of breast cancer compared to people who are unexposed or who have less exposure. If you have a risk of 1, it means you have no increased risk. Your risk is 0. Not you have 0 risk of getting breast cancer, but your baseline risk or your reference risk is 0. If your relative risk is 1.5, it means you have 50% increased risk of getting breast cancer if you have been exposed to these chemicals. And what we found is that women who used these 4 chemicals had a 40 to 50% increased risk of breast cancer associated with the use of these chemicals. Now, one of the things that we try to do in epidemiology is to look for internal consistency to give us more confidence in the results that we get. And one of those tests is to look also what risk there is associated with the husband's use of the chemicals. For this, we found that only that the husband's use of turbofos was associated with a woman's increased risk of breast cancer, but not the other chemicals that the women reported using themselves. So we're still trying to sort that out. But that's an ongoing question. Another aspect of this, in terms of looking at internal consistency, is that we found similar results across the 2 states. So we're getting similarly increased risk in both Iowa and North Carolina, which again increases our confidence in the findings that we're getting. I want to go just briefly through some of the strengths and limitations of this study, because it's important to understand the challenges of some of this research. first of all, we're looking at a lot of different associations here, and one of the things that we talk about in epidemiology is false positives. The more tests you do, the more tests that will be statistically significant by chance alone. So we use other mechanisms to try and address that point to again increase our confidence that the results that we're getting are real. But some of the other challenges are that we had limited data on the women's use of the chemicals. We don't have data on their early life exposure because those questions were not originally asked. And these data are self-reported, which is both a strength and a weakness of this study. So in general, we try to come up with objective measures of exposure so that we can more accurately estimate or assess what a risk is in a given relationship. There really is no good way to go back and look 20, 30, 40 years at what someone's exposure is to these chemicals without just asking them. So this study is one of the few that has done this, and so while it is a limitation, it also is a strength because it's really one of the only ways to do this well. But importantly, these exposures were assessed before diagnosis, which is, again, a bias that we look for in epidemiology. So these are pre-diagnostic information. We have a large cohort, we have a wide range of exposures, and we have excellent cohort follow-up. And I'm gonna skip over that and go on to the next slide and talk about another topic that I have been studying for a number of years, which is polychlorinated biphenyls and non-Hodgkin's lymphoma. And I use this example because it poses a different set of questions, a different set of challenges than the pesticide study that I presented previously. So polychlorinated biphenyls, also known as PCBs, have been around since the late 1920s, but they became much more common and much more widespread in the 1950s and 1960s. Levels peaked in the 1960s, 1970s, before they were banned in the United States in 1976. But they're ubiquitous in the environment. Even today, even though they've been banned for about 50 years, you still get detectable levels in our bodies. You get detectable levels in the environment because they're extremely persistent. And there are, we talk about PCBs, again, like pesticides as one thing, but in fact, it's a family of chemicals. And this is important because the chemicals are all structurally different, and so they have different chemical and biological properties. It's important to recognize when we're looking at what their possible health effects might be. Now, historically and still today, most of the exposure to these chemicals comes from our diet, especially animal products, so fish, meat, eggs. These chemicals tend to bioaccumulate, and so we tend to find these primarily in animal products. So the purpose of this study that I'll be talking about now was to look at the relationship between PCBs and non-Hodgkin's lymphoma in three different prospective cohorts. These are cohorts that in which people had been enrolled and blood specimens had been collected, and they were followed for cancer and other health outcomes after that. And those three studies are the Janus cohort. Well, I'll talk more about those in a minute. The first one is the Janus cohort, which is a cohort of Norwegians who were enrolled in the mid-1970s, and blood samples were stored, and we went back and looked for cancer incidence in this cohort, and we identified 194 cases of non-Hodgkin's lymphoma in that cohort. So we pulled their samples. We pulled 194 control samples, people who had not developed non-Hodgkin's lymphoma in that cohort, and we analyzed these for PCBs. The CLU-1 cohort was a cohort out of Western Maryland. It's a smaller cohort, but we identified from this cohort 74 cases of non-Hodgkin's lymphoma, and also pulled a number of controls from that study. These samples were collected in 1974, and I give the years because it's important. Again, as I mentioned a moment ago, the body burdens of these chemicals peaked in the 1960s, 1970s, and started to decline after that, because it's relevant to the last study, which is the Nurses' Health study. What I'm presenting here is some pilot work that we did at the time that this study was initially done. The specimens from that study were collected in the late 1980s, so levels are already on the decline in that population. And that study was relatively small. We had only 33 cases in the nurses' health study. Now, what relevant for the next part of my presentation is that we measured a number of different PCB chemicals, what we call congeners, in these samples. And we also measured a number of pesticides, including DDT, although I won't be talking about DDT or the pesticides today. I'll be focusing on the PCBs. Now, this slide's really complicated, so let me walk you through it. I'll try not to fall off the podium. So first, generally speaking, what this slide is trying to illustrate to you is that increasing concentrations in your blood of different PCB congeners are related to an increased risk of non-Hodgkin's lymphoma. That's the big picture I want you to take home from this. But the way we get at that is as follows. So again, as I mentioned in the previous slide, a risk of 1 means it's your baseline risk. You do not have an increased risk. Everything is relative to that. On this axis here, we're looking at what your relative risk is. So if your risk is above 1, it means you have an increased risk of non-Hodgkin's lymphoma. As you go across, what we have is increasing levels, 1, 2, 3, 4. Those are increasing levels of specific PCB congeners in each of the cohorts. And I've shown, looking back, is a three-dimensional plot. Looking back, I show each of the 3 different cohorts that we studied. The one in front, the one that's kind of purple, is the Janus cohort. The kind of maroon one in the middle is the CLU-1 study, and the one in the back in kind of lime green is the nurses' health study. And the point I want to make here is a couple of things. One is that you will notice that risk increases as your blood levels of these chemicals go up, your risk goes up of non-Hodgkin's lymphoma as well for these particular congeners. And that while the magnitude of the risk varies across the different studies, you see a pretty consistent pattern in those, in each of the cohorts of increasing risk with increasing exposure to these, or increasing body burden of these chemicals. Now, we did not see any trends for the other PCB congeners that we studied, which is why I'm focusing on just these 3 here. All right, I'm going to skip that slide in the interest of time. What I do want to show you here is just, again, that to show you how IARC has classified organochlorines, many of which we have studied, is that they have identified a number of organochlorines as established group 1 carcinogens, and I've shown these here, including PCBs. And also, it shows the cancer for which they've been definitively linked in the IARC classification. And again, I can share all this information with you later. So just to bring this to a conclusion, I want to just note a couple or a few points. One is that epidemiologic studies of environment and cancer are quite challenging. And again, this isn't to despair, but just to point out that the challenges that we face and why progress has been slower than we would like it to be. But nonetheless, we are making progress. We have identified a lot of carcinogens already. There's a lot more research being done to identify and strengthen the evidence for others. Exposures are often low in the general public compared to other occupational groups, farmers or industrial workers and so on. So it poses certain challenges in studying these. And it also means that our risks are relatively lower than it is for farmers or others who are more highly exposed. But the number of people affected is substantially larger. So what we call the population attributable risk can be quite substantial even when our absolute risk for any one of these exposures might be quite small. As I've noted, these exposures can be difficult to characterize or to quantify just because of the time lags involved and because of the way that we interact with our environment and interact with these exposures. And in addition, some of the emerging exposures that are of particular interest to us today, things like the perfluorinated chemicals and microplastics, are receiving a lot of attention, but there are some unique challenges to studying these, especially as the exposures change. So in the case of perfluorinated chemicals, going from 2 to going to thousands in a very short period of time. So it complicates the efforts to do the research needed. However, there have been a number of developments in recent years that are greatly advancing our ability to study these questions in a more systematic way. And this includes development of methods and technologies to do more high throughput testing. This largely consists of in vitro testing, where we can now test hundreds or thousands of chemicals simultaneously. We can administer a number of different tests, in vitro on these chemicals. And so we're now getting to a point where we can do some of the things that previously we had to do one by one and in a very slow and time-consuming way. So I think that the rate at which we'll be able to address some of these questions is greatly expanding. However, I want to also emphasize that this isn't a panacea. This doesn't solve the problem, because these in vitro tests, these high-throughput tests, don't address all of the questions. They tell you what's happening in a petri dish, but they don't tell you what's happening in the human body. And to the extent that they mimic what happens in the body, they're effective tests. But to the extent that they diverge from what's actually happening in our bodies, they're imperfect. So they can produce false positive results, they can produce false negative results. There's still a major advance, and we need to keep going in that direction. But the animal tests can't be rushed. The human epidemiologic studies can't be rushed. These studies just take time. And I think I'm going to stop there. And then just go back to this slide again, just to remind you that, despite the challenges that we face and the obstacles that slow us down and make our work more difficult, we actually have made a lot of strides. And we have identified a number of chemicals that we had, and other agents that we are in the process of either removing or reducing exposures to. So I think that there has been a lot of progress, and I think that, with some of the new technologies coming along, we'll make a lot more progress in the coming years. And I'll stop there.